There you go.
All right. Thank you so much, Dushin, for this opportunity to present in the Pharma Pro series. Is always super exciting to be here and learn about the diverse things happening across campus in terms of pharmacy and invention. I was playing some music just before, and so to those of you in the chat, yes, that was intentional. We love to think of turning molecules into music. It's a program that we have. I won't get a chance to talk too much about it, but just imagining that music is information, molecules are information, and trying to bridge the gap between those two types of information is something we do with machine learning, and it's a lot of fun. And so it's fun to make music. It's fun to make molecules in that way, and I'd be happy to talk about it more. But I'm here to tell you about generating drug structures using machine learning and flasks, and hopefully I succeed in getting through this presentation. I'm a synthetic chemist. I'm a synthetic organic chemist, and I like to think about making molecules. I think it's really important to think about how to make molecules. It's a very old technique.
Can you ask online to see if you can hear you all right?
Can you hear me online all right? Can someone
respond? Some online, some online. Yes.
You can hear me? All right, splendid. Thank you. All right, all the AV is settled. We're ready. Making molecules is really important. It's where our drugs come from. If you can't make them, you don't get your drug. And so thinking about where drugs come from, I think requires a lot of discussion around their synthesis. And this is something we've been studying for a long time. And it gets to piggyback off of a centuries-old field of synthetic organic chemistry. And so we get to think about how molecules were made hundreds of years ago, or at least 150 years ago, and how they're made today. But we design molecules in the computer. We'll talk a little bit about that today, and then we make them using chemical synthesis. And if you make them, you get to test them, and hopefully you learn from that testing a bioassay, an in vivo assay, an in vitro assay, if your design was a good one. And so you likely have some hypothesis. If I switch this oxygen atom into a nitrogen atom, I might get a better pharmacokinetic profile. I might get a molecule that binds more tightly to the protein inhibitor or protein structure I'm trying to inhibit. And so the making aspect of this design, make, test wheel is absolutely critical to the process, because it influences what you can design. You're not going to design a molecule that takes 5 years to make. You might, but you'd have to be a pretty brave synthetic chemist to want to invest that type of time installing complicated functional groups and stereochemistries in just the right way. When I started my career, it was in the pharmaceutical industry, and so much of the synthesis I was involved in was being outsourced. There was this notion that we could just buy it overseas, and it wasn't necessary to really perform synthesis in-house anymore. And so I spent a lot of the earlier part of my career asking the question, does synthesis matter? Like, do we even need synthesis anymore? And I've pushed pretty hard that the answer is yes, and hopefully I can try to convince you. But there was one really poignant story that I heard in a podcast last year from the CEO of Moderna. And he articulated that the genome of the COVID virus was published on January 11th of 2020. And on January 12th of 2020, Moderna had their first physical sample of the COVID vaccine in hand. 24 hours and you had the drug ready. There was obviously a lot of development that came after that, but he framed it entirely through the light of synthesis, right? That if it were a small molecule medicine, it would have taken years to develop, or at least a year to develop, as we saw with Paxlovid. But with the COVID vaccine, they knew exactly how to make it, because there were 4 building blocks and there was 1 reaction. There's like 0 choices to be made, right? So there's no decisions to get lost in. There's just, these are the building blocks, ACTU, and these are the reactions we're going to use to stitch them together. And so I think that synthesis enabled the production of the vaccine, or the invention of the COVID vaccine, instantly or near instantaneously. You might say, well, all right, let's just have mRNA vaccines be the future of medicine. And I think they got a bright future in medicine, right? But where did the conversation go to as soon as we got the vaccine in hand, it was like, well, how many -80 freezers are there in Bangladesh? Or like, where are we going to get -80 freezers to store all of these vials? And what's the shelf life of these mRNA vaccines? You don't have that problem with small molecules, right? Small molecules you can just dump at the back of a plane, and they're gonna, you know, they last, they're shelf stable. They get inside of cells, and that's relevant. So we focus in my lab on small molecule chemical synthesis. I think we're bit in pretty hard to the idea that synthesis does really matter, and that, you know, that synthesis enables the invention of medicine through this design, make, test wheel. And so for today's discussion, I wanted to talk about what are molecules. We were listening to some music earlier, and we like to talk about how molecules can be music. Molecules can be lots of things. Molecules can be an idea. Molecules can be a mathematical graph. Molecules can be a chemical structure that some might view as simple, some might view as complex. What are drug molecules is just, you know, a subset of molecules. I don't know how much justice I'll do to the question, what is artificial intelligence? But I'll show you a tiny bit of machine learning, and then spend, if I get there, enough time talking about how AI and automation can generate molecules today and in the future. So, all right, so what are molecules? So here's a molecule, right? This is a line representation of the molecule benzene. And so we can talk about this as being described as a hexagon. It has 6 carbon atoms that are arranged in this way. And down on the bottom is what's known as a smile string. This is a text representation that computers can understand of this chemical structure. And so hopefully most on the call will have seen a molecule of this type before. The notion of drawing a hexagon to be a molecule is hopefully something that we're familiar with. But there's 6 carbons and 6 hydrogens to this molecule, and you can see it represented as this 6-membered ring. You can see it represented in this kind of funny way that, you know, is a little hard to articulate or to see as a human, how C1 equals CC equals CC equals C1 represents this, but a computer understands it pretty well. Here's some more sophisticated molecules. So these are diverse antiviral molecules. We were exploring the synthesis of these during the COVID early days. And I think they're interesting molecules to look at. One of the things that I hope will jump out of the slide is diversity, right? These have interesting chemical structures. They look different than the benzene that I showed you before. They do different things. Here's oseltamivir, which is a neuraminidase inhibitor that is, you know, potentially very interesting for treating avian flu. And lopinavir and remdesivir was, of course, one of the early darling childs of COVID treatment. You know, they have these chemical structures that are these line drawings. So you can see there's, you know, the lines represent different carbon-carbon bonds. And if there's a line to an OH, then that's a carbon-oxygen bond. And so that's one way that we can think about it. If we studied organic chemistry, that's probably our comfort zone. We might just call it by its name, oseltamivir, lopinavir, remdesivir. I can go to a pharmacist and say remdesivir, and they'll have a sense of what I'm talking about. They know, you know, they have a feeling of like, what is remdesivir? If I'm talking to a computer, I'm going to put it into this smile string as shown here. And so there won't be any quizzes on this. We don't need to know exactly what smile strings are, but I want to give you the sense that these 3 different ways of depicting this molecule are all saying the same thing. And so different computational programs will handle them effectively the same. And you know, computers can understand that if I type in the word remdesivir today, a computer can rapidly match that to this smile string and then rapidly draw this chemical structure in this way. I would call these antivirals complex. These are small molecules. They're much smaller than a vaccine, than an mRNA or a biologic. But I would still call them quite complex. If my student said, you know, I'm going to go and make this, I would think, you know, we'd be embarking on more than a couple days of work to make one of these molecules. These are sophisticated molecules. They have bonds that are drawn as these wedges and dashes, meaning that there's 3-dimensionality to them. And so we stitched these together with multi-step synthesis. We call this total synthesis in the field. So we would make one of these molecules. Here's a depiction of a synthesis of oseltamivir, drawn as these stick and ball models, but actually these are meant to be mathematical graphs, and so we won't get too deep into graphs today. But if you study graph theory in mathematics, you can plug molecules directly in. And so hopefully what you can see is that oseltamivir here is also shown down in the bottom left corner. And you can see that it's kind of being built up by these sequences of reactions. So there's 3 reaction arrows, and they go through different reaction steps. Step 1 brings some of these pieces together, then step 2 and 3 clicks more of the pieces together. Steps 4 and 5 kind of finishes up the molecule. And so some brave chemist embarked on a 5-step synthesis of oseltamivir under a hypothesis that oseltamivir, that this exact chemical structure was a good one. There wasn't, there wasn't proof because it was unknown, and it had to have been unknown because the company that invented it wanted to patent it, right? So we have to be exploring the unknown, or else we're just borrowing someone else's intellectual property. And so that means you're designing new chemical structures, then you go and make them. Nature gives us molecules like this. And so these are synthesizable molecules. These are very complicated molecules. Many of them were synthesized through the efforts of perhaps a dozen PhD students. And so these are very difficult molecules to make, but using modern technology, it's possible to make these. These are natural products. We tend to like going to look in nature and finding complex molecules to attempt to make. Here are some drugs. These are modern drugs. These are, so sotorasib came out, I guess I can't remember, it's late 2021 or 2022. But for those of you that have seen this molecule before, recognize this as an absolute breakthrough in cancer therapy. This one is quite strange in that the benzene ring down on the bottom here, do you see these 3 bonds are drawn to be kind of bold? That means that there's a very strange locking of 3-dimensionality in this particular molecule where the methyl is coming out of the plane of the page and this isopropyl group is going back into the plane of the page. And that's really, really, really difficult to make synthetically. But through advances in chemical synthesis, something like that is now on the table. MK0616 is, I mean, look at this beast of a molecule. So synthetic chemists at Merck embarked on the synthesis of this. Someone said, I think that's a good molecule. It should bind tightly to PCSK9. Let's go and make it. And you can imagine that it took some people a good amount of time to figure out how they were going to piece that particular molecule together. Here's a new one from this. I just pulled this from Drug Hunter's Molecule of the Month today for 2023. This is a really cute little CDK2 inhibitor that's got this macrocyclic ring structure and several stereocenters. Got a 4-membered ring and various heterocycles. And it would have been impossible to propose this molecule, QR6401, or any of these molecules 10 years ago. Because I mean, these are molecules that maybe could have existed 10 years ago, but they would have just been too impossible to realistically propose as synthetic targets. Advances in synthetic chemistry make it possible to access structures of this complexity today and to have them be the type of thing that we want to go and invent. All right, so I've already shared a little bit about drug molecules, but let's go a bit further back. And so I had the great luxury of visiting, I'm gonna have a trick here. They want me to call it Merck, but I'm on American soil, so it's not Merck, it's Merck KGaA, or Millipore Sigma, and I do receive funds from Millipore Sigma, but I went to their 350th birthday party. Merck was, I just can't call Merck. I worked at Merck & Co., and I'm trying to talk about the German Merck. They used to be one company, and a young boy left Germany to build a Merck in Manhattan, and when World War I broke out, they had to be separated. So there's a Merck & Co. in America and a Merck KGaA in Germany, and I get myself into trouble with those names all the time. But I went to celebrate the 350th birthday of this company, and a source of enormous pride for the company was that they were the first pharmaceutical company to market molecules. That prior to their discoveries in the 1600s, they would sell. This is what medicine looked like, right? It was a notion of which types of herbs and mushrooms would cure your disease, and so Merck in the 1600s started to isolate molecules and got pretty advanced over the next several hundred years. In the 1850s, they would start selling distinct purified molecules coming out of these plants. And so I show here, you know, an old molecule, but you can imagine the transition of like going to your pharmacist and getting a poultice or a you know, a bundle of flowers into a bottle of here are chocolate coated strychnine tablets, which I think is a really intriguing formulation. But you know, at some point, about 100 and what are we, 2023, like maybe 140 years ago, we started to sell independent molecules, purified molecules, instead of just plants and fungal broths. And so at that point, you know, there was this notion of a chemical structure, and where can we go from there? And so so today we would talk about the invention of discrete molecules through this design, make, test cycle to make single molecules that have a function. You know, you might like to think of them as looking like this. I think that this is one way to think of our objective as navigating that design, make, test cycle. I like to think of it like this, where we are steering towards a population of molecules that satisfy multi-parameters. So let's imagine that a drug molecule exists somewhere in a property space or in a experimental space as this black dot at the center of this Venn diagram. And so if we want a nice medicine for our customers, for our patients, it should likely have a balance of properties. If we want to have a once daily pill, which is a very convenient way to take medicine, we would want to have something that is soluble, so that when we swallow the pill, it dissolves in the aqueous layer of our tummy. We probably want it to be permeable so that it gets across the organic layer of our intestinal cell membranes. And then that will get it into the aqueous layer of the blood, where it will, you know, go find its way to the liver and need to deal with its metabolic stability. We should like most molecules to have the right balance of metabolic stability so that we're ready to take our next pill 24 hours later. That's a very convenient dosing regimen for patients to deal with, and so for most medicines, that's what I like to think of. Of course, if you're working with a sleeping drug, you want a much shorter window. You don't want to be sleeping for 24 hours. And, you know, maybe you want to talk about long-acting drugs, and I'd be happy to talk about that. And we think about other properties like selectivity, and then, of course, potency. Here I'm alluding to, does it bind to the protein target that we believe is associated with the disease? Now, I'm a synthetic chemist, and I made this slide, so I get to draw a great big circle of synthetic accessibility and point out that chemical synthesis favors certain types of properties. And so, you know, we dream of reactions that are easily able to access this exact pocket of chemical space where molecules are selective, soluble, permeable, potent, and metabolically stable. But we do a lot of our reactions in very lipophilic solvents like dichloromethane. And so, you know, we kind of tend towards lipophilic molecules just because synthetic chemistry is kind of born of the petrochemical industry. So I want to bring us a bit up to the minute and think about how modern drug discovery is performed. We have this design, make, test cycle of invention, right? We design a molecule. I've been trying to convince you that it's important that we can then go into the lab and effectively make it in a short order of time. And then we test it by whatever bioassay is relevant to the problem at hand. That could be a purified protein binding assay. It could be a cell assay where we're looking at the phenotype of the cell, or it could most certainly be an in vivo assay. But there's this design phase that we need to think about. What are all the molecules that are out there? And there's a notion that there's an infinite number of molecules. People like to use the number 10 to the 60. There's, you know, the notion that there are more drug-like molecules that can be imagined than there are atoms in the universe. I don't know if that's a reasonable, you know, I've heard it said, and anyways, there's a lot. We can't make them all. We can't make anywhere near a tiny fraction of the molecules that we can dream up. So we need to design them. And so today that would typically look like identifying a target structure, a protein that we believe is associated with the disease, and imagining how the small molecules that we think we can make would bind into it. So I pulled up an image from the landmark 21 paper on AlphaFold, just showing that it's possible to predict the protein structure or the structure of a protein. So AlphaFold is a algorithm based on all of the crystallographic data that's in the PDB database of protein crystal structures. And so on the basis of that available data and many years of research, the modern ability to predict protein structures is quite good. And so AlphaFold is one popular way of doing this. There are certainly others. And so you can see that the green is the experimental, and blue kind of looks about the same. So if we know that a patient has a disease, they've got, we're trying to tackle, someone with a funny mutation in their KRAS gene, we can generate a protein structure. Ideally, we've done it experimentally, which would be the green one. But today in 2023, if we don't have that, we can get a decent, starting point with a program like AlphaFold. And then, so what we would then like to do is, now we've got our target, we want to stick to this with a small molecule that's going to inhibit its function in some way, or perhaps agonize it. And so I pulled this fun graphic from this is from today's issue of Nature. There's a great review on computational chemistry in today's Nature, and this graphic I think spoke to me a lot, where they show that if you are able to propose more chemical structures in the computer, so virtual chemical structures defined here as a bigger chemical space, you improve your chances of finding low nanomolar or low binding hits. Keeping in mind that for a pharmaceutical, we'd like to be in the nanomolar range, ideally, low nanomolar range. And so if we dream of chemical structures, small molecules that we think we can go and make, we could do what's called docking in the computer, where we would stick those molecules to the active site of this protein crystal structure, or protein predicted structure if we're using the AlphaFold model. And what we would like to do is take molecules that we think we can build quickly, stick them in, and pick the best ones. Computers cannot yet say this molecule will with 100% certainty be a 1 nanomolar inhibitor, but we're actually pretty far from that goal. But we're still able to predict the binding of small molecules to proteins in some sense. And I'd like to point out that we will soon be joined by Tara Stein, is a new professor joining MedChem, and she's going to be we're very excited with what Tara is going to bring to the community in terms of looking at protein structures and small molecule structures. But so there's this notion that large chemical space is better, because the more chemical space you can evaluate, the better chance of finding a better compound is. So here, if we look at 10 billion compounds, we've improved our odds of getting down to that 100 nanomolar starting point. We can imagine mathematically 10 billion chemical structures. I may talk a little bit about that, but it's only very recently that computationally it's been feasible to look at such large chemical spaces. And so the advancement of computers has really enabled huge advances in the invention of medicine. So, all right, I gave myself the task of presenting under the topic, what is artificial intelligence? And I put together my slides, and I don't feel confident that I'm going to answer this particularly well, but here's what I want you to know. So what is machine learning? All right, so artificial intelligence is just like a bigger umbrella of machine learning. And so I'm sure that a number of people in the audience have heard of ChatGPT, and we'll talk just a little bit about it. But the only thing that I really hope you pull from this talk is that machine learning can be built from data, descriptors, and an algorithm. So when someone says, I did machine learning, like, it sounds fancy. And if they call it artificial intelligence, it sounds even much more fancy. And so it's probably right. But, but it's, there's a good chance it's not right. And And all that they mean when they say I did machine learning is that they took some data, they appended descriptors to that data, and then they performed an algorithm on it. And so if you have used Excel and done an XY scatter plot and put a line that fit that XY scatter plot with an R squared on it, you have done machine learning. Okay, it's not super fancy because it's only two-dimensional, right? So the descriptors that went into that function would have just been your X and your Y values, whatever they were. Maybe it was, temperature outside versus ice cream sales. And then those would be your descriptors, and then you fit a line of best fit through it. It's, probably a lot of people in the room or on the call have done that type of machine learning where you have two-dimensional data and you fit using an algorithm a line through the data to get an R squared. Now, that's not fancy enough to call it machine learning. So you maybe if you were cool in school, you did a three-dimensional version, right? You made a 3D plot and you showed a line coming out in 3 dimensions. But we can think of things as having many more dimensions, right? It's like describe Tim in I don't know, X, Y, Z coordinates and like what is it that makes up a human? There's lots of ways to describe that, right? So I'm sure a lot of people have heard about ChatGPT 3 versus ChatGPT 4. And my understanding is that the difference between 3 and 4 is that ChatGPT 3 is based on billions of descriptors, where ChatGPT 4 is based on trillions of descriptors. And of course, the data that goes into the ChatGPT model is the internet, right? So we have all the images on the internet, we have all of the text and words that are on the internet. How do we describe what those are, right? Like you can take a picture of a cat off the internet, you can describe it as like pixels that have this X and Y position, and they're based on these different colors. But if you took a picture of a cat and imagine describing it with a billion different descriptors, it's a cat, it's cute, it's got a pixel that's orange here and a pixel that's yellow here. And then if you described it with trillions of descriptors, you'd get like a more, I don't know, maybe more of a notion of what it is. And then there are very different algorithms that one can use. And I'll talk about some very simple ones in the coming slide here. And so I talked about, here's what you've done hopefully before. You've fit some two-dimensional data. So here's like an XY scatter plot that's got a line going through it. And so we can imagine data that is higher dimensional than this. So let's imagine data that can be described in 10 descriptors. What that looks like is you have an Excel spreadsheet with 10 columns of numbers in it, right? And so there's your 10-dimensional data. You can't draw easily a line through 10 dimensions in your mind, but a computer can do it. A computer can navigate through those 10 dimensions, those 10 different columns in your Excel spreadsheet and find a line of best fit through all of them. And so that type of algorithm is a very simple one called principal component analysis. It's very commonly used to cluster data. And the beauty of principal component analysis is you take very highly dimensional data and reduce it down to typically 2 dimensions. So I show here the drug bank database shown on principal component analysis. So the drug bank database has several thousand, about 10,000 drug chemical structures in it. We would have loaded them in as their SMILE strings. And in one instance, we scored them by their physical chemical properties, things like hydrogen bond donors, molecular weight, partition coefficient or log P, hydrogen bond acceptors. And then we had, I think, 13 descriptors, and we condensed them. And then using the PCA algorithm, did this 2-dimensional plot. We took the exact same set of molecules and fed them through a fingerprint algorithm that turned our data into 2,000 dimensional data. And then we put it through PCA, and it gives us, again, this 2-dimensional data. The point that I want to make here is that there are 4 drugs: Januvia, Gefitinib, Rapamycin, and Navitoclax, and they're colored differently in the plot. And what you can see is that the descriptors, whether I chose my 13-dimensional physical chemical property fingerprint or my 2000-dimensional ECFP4 fingerprint, influence where these molecules are, right? So let's imagine that we are running an HTS or a drug screen. We get a hit, it's killing the cancer cells that we wanted it to kill, and it's this blue dot here. So if I had clustered on physical chemical properties, I might say, well, I want to go buy molecules that look like that one. I'll get this red one because it's kind of close to that, right? But if I had chosen to cluster on a completely reasonable other set of descriptors, these ECFP4 ones, if the blue dot were my winning hit, I might say, well, actually, I'm going to buy the green one because that one's closest to, right? So I just want to give you the sense that if machine learning looks at data, descriptors, and algorithms, the descriptors have an influence on what your output is going to be. Here's a similar story borrowed from my friend Pat Walters at Relay. This is just the same data, the same descriptors. I can't remember how many drugs it is. Looks like a couple thousand. These are actually the blue dots are virtual chemical structures that were imagined, carbon and hydrogen and oxygen bonds in the right way. And the red dots are ones that were tested in a bioassay. And so we have experimental data for red dots. We are looking at virtual chemical space with the blue dots. And we're going to use two different algorithms to cluster them the same. They were both described using molecular fingerprints. So they were using the same set of descriptors. With principal component analysis, you can see that the actives kind of there's one cluster here and then kind of a scatter shot of actives around. And so that's what principal component analysis would do to this data set. That's that algorithm. There's another very common algorithm that people like to use called TISNE, or T-distributed stochastic neighbor embedding. And you see it kind of makes the clusters a little bit more tight, or at least this cluster becomes a little bit more tight. So exact same data, exact same descriptors, different algorithm. And imagine you're looking at this data and trying to infer a decision, right? You can see how different algorithms might influence your decision, how different descriptors might influence your decision. So the modern world looks like we're in a world where we can generate a protein crystal structure and imagine it's associated with disease. We want to go enumerate new chemical structures that we're going to, in the computer, stick into this protein structure. And if we think that we found some good ones, we're going to go and make them. We need to make sure that they're easily accessible enough so that we can make them on the timeline that makes sense to us. Maybe I need to have my result by next Friday. I'm going to pick something I can make in 2 weeks. If I can be a bit more brave and wait a little bit longer, it'll take longer. But chemical synthesis is hard, and we're deeply invested in this problem. And so for us in the Cernak lab here in Ann Arbor, we love to set up big flasks and stir stuff around and mix purple powders and white powders and hope that we get the right chemical structure that we wanted. We need to do that using things like catalysts and reagents. And so typically we'll put a little bit of copper and molybdenum or manganese and in various forms. We have many thousands of metal salts in our lab that can be added into these pots to try and stitch together the different building blocks that we can also purchase. We tend to think of it like building Lego. So you have these little Lego blocks that you can stitch together. And then we use Python and various tools to navigate this. Here's just some more, we are an experimental lab and a computational lab. And so we love to do experiments. And so I want to share here some of the chemical reactions that have gone into what is now a $1 trillion pharmaceutical industry. Multiple groups have repeated this result. Unfortunately, I can't see the references here. I don't know if those online are able to see it. But there's a reference down here from Jonas Bostrom. And so what he did was he scraped 40 years of papers from the Journal of Medicinal Chemistry and came to this conclusion. You'll get a nearly identical pie chart if you scrape the entire US PTO of patented pharmaceutical preparations. What you'll find is that in this $1 trillion pharmaceutical industry, we gravitate towards a small subset of chemical transformations or chemical reactions. The amide coupling is the top of the list. So that takes 1/4 of the pie. And then this is followed by this Suzuki coupling, is this carbon-carbon coupling, and a Buchwald-Hartwig coupling where we take this hexagon with an X on it, which would be a bromine or a chlorine could be perhaps an iodine or a fluorine, and then a nitrogen that sticks on. We make a carbon-nitrogen bond. So those three reactions make up the large majority of what we would imagine running when inventing new chemical reaction, new drugs that would then bind into those protein crystal structures. We've done a lot of work while I was working at Merck on the CN coupling reaction and miniaturizing this. And we've also recently done some work on some of the other top reactions in the pie. Suzuki coupling, reductive amination is this kind of dark green wedge, and then Bach protection. And so this has very recently been accepted for publication in the journal Nature Synthesis. And here we're able to do the Suzuki coupling, a reductive amination, or this N-alkylation and Bach protection using this robot. And the reason this robot is important to us is because it enables us to run lots of chemical reactions. And so hopefully I've given you the sense that there's so many chemicals we need to consider. We can use algorithms to help us navigate through all of this data. And we are interested in finding new chemical reactions that will enable the hunt for drugs and speed up the invention of the medicines of tomorrow. One thing that's important with these particular reactions is that they're homogeneous. And so here is these reactions run in test tubes. You can see that they're clear liquids. And so that's important because we do all of our work, at least in this automated format, using liquid handling robotics. And so things need to be liquids if they're going to be handled by liquid handling robotics. And so it's actually quite challenging to find reactions that are nice and liquidy, because keeping in mind that we use lots of manganese and molybdenum and copper and iron, and these things, they don't really care that I wanted to use a robot that only likes to work with liquids. But the biggest chunk of the pie is the amide coupling. And so in launching our labs at the University of Michigan, the plan was to ask the question, why is this the single reaction that makes up 25% of a $1 trillion pharmaceutical industry? Why is it that if I'm presented with an amine and a carboxylic acid, I would always choose to unite them to make this chemical substructure? There's some ramifications to making this chemical substructure. It's got an NH on it, so it's got a hydrogen bond donor. If you are trying to make a medicine that's going to get into the brain, you don't want to have very many hydrogen bond donors. And so it's interesting that, in this $1 trillion industry, we would take an amine acid, stitch them together to make an amide. That reaction just gave you a hydrogen bond donor. So, by running this reaction, you've lowered your chances of getting into the brain. I show here a methyl group and a methyl group as the jewelry attached to the reacting atoms. You can of course go and, this is not a drug, right? You're gonna have other pieces on there that would have been designed to sit inside of the active site of that blue or green protein crystal structure or protein alpha fold structure I showed. But regardless of what the jewelry is, this methyl group is a big heterocycle that sticks right onto the protein in just the right way. We've just united it to the other piece that brought us the nice solubility and pharmacokinetic properties that we wanted using this amide coupling, and by virtue of using the amide coupling, we've made the thing less able to get into the brain. So why is this the single reaction that we use most frequently in the pharmaceutical industry? It turns out that there are many other ways that you can imagine bringing these molecules together. We have shown in our labs that if you take this ethylamine, this NH2CC, this carboxylic acid, this oxygen C, oxygen CC, take those 8 heavy atoms, throw them in the air, let them land with the only rules being that carbon gets 4 bonds, nitrogen gets 3 bonds, oxygen gets 2 bonds. There are 56 million ways that these can be brought together, and yet we choose one, right? So it's interesting that in the pharmaceutical industry, we choose just one chemical transformation to unite them. And if I want to change the properties of my molecules, I say, go to our big box of Lego, which is the chemical vendors, which I can buy the pieces from, get new pieces of Lego and bring them in, and we'll stitch them together with the amide coupling. And where we said, what if you only had 2 pieces of Lego? What are the other ways you can stitch them together? It turns out there's 56 million, and each one of them influences the property of the molecules. So shown here are two building blocks stitched together with different catalysts, and we're able to stitch something together with the classic amide coupling shown here, and we have that hydrogen bond donor. We have a boron catalyst that reduces it down to make a basic amine. We've borrowed conditions to make this using an iodine catalyst to make these really aromatic and rigid compounds. Using Goossens conditions, we can form an acidic compound, and if we work up those conditions with copper and heat the bejesus out of it, it will decarboxylate and give us this very lipophilic compound. And then Yuning Shen, who I'm so honored is here with us today, has very recently graduated from the group and invented this esterification. And so, the cool thing is that we're changing the properties, whether this thing is lipophilic, polar, basic, acidic. I'm changing that because I chose copper, iridium, iodine, or iron as my catalyst. And that's kind of neat, right? That's a different way to like turn a knob over here and it influences the properties over here. And we're exploring what that means. And so I'm so excited that Yuning is here today. Yuning is our first graduate student. I had the luxury of hooding her this morning, and so that's why I have mortarboard hair. And so Yuning is the first person to graduate from our group, and so she also discovered the first reaction in our group. We use robotics and high throughput experimentation to run lots of reactions, navigating through chemical mixtures of copper and trying to find catalysts that stick things together. And so Yuning was interested in finding a reaction that would take an carboxylic acid and an amine activated as these diazonium salts and then put it on a robot. And we were able to run lots of chemical reactions and make lots of diverse analogs that are not amides. And so this was a pretty exciting win for the lab earlier in our days. Yuning is, this was the luxury of working in the College of Pharmacy is that we had a lot of access to pills that we hadn't thought of. And so Yuning came to me one day and said, hey, I got all these yields on these drugs. And I said, where did you get these drugs? Like, those are expensive. We can't like go buying expensive drugs. She said, no, I went to Walgreens. They were very cheap. They were like $2 each. And so we, by being a synthetic chemist in pharmacy, we get to have a lot of fun. And so we have been exploring this notion of reaction enumeration and trying to navigate to these 56 million unknown chemical reactions. Each one can influence the properties of the drug. Can we invent these new 56 million reactions? We have this robotic platform that we developed first at Merck and now is been rolled out to many pharmaceutical companies. And so we're able to very rapidly invent new chemical reactions. Here's just a snapshot of some of the achievements that we've had in the lab for the invention of new amine acid cross-coupling reactions. And so keeping in mind that, in a $1 trillion industry, there's a single chemical reaction that one would imagine for uniting an amine and an acid. It's to make an amide bond. And we're now showing that you can make carbon-oxygen bonds, whether it be on aryl groups or alkyl groups. We can make carbon-carbon bonds, whether it be on alkyl or aryl groups. We can install a ketone. We can make aryl-aryl CC bonds. We can make CN bonds, and we can make reduced CO bonds. And so we've been having a lot of fun exploring this. It's incredibly difficult to discover a new reaction. We have been very generously gifted funds from Schmidt Futures to advance the amount of data available for chemical synthesis using transition metals. And so it's our goal to publish 250,000 chemical reactions for the community to do with whatever they want. We intend to invent as many new chemical reactions from this data as possible, but it is our dream that people who we have never met before will come to the data and invent new machine learning models to help us invent the next chemical reaction. And then that chemical reaction could be the one that hopefully helps to invent the next cure for cancer or Alzheimer's or diabetes. We do a lot of this with a software that we have written in-house called Factor. And Bo Majer is an incredibly talented student in medicinal chemistry who had the idea on a Monday to plug Factor into ChatGPT. And by Sunday, we had submitted the paper to ChemArchive. And we learned just last night that Organic Process Research and Development has done a highlight on this particular ChemArchive paper, which for us is a huge honor. That's OPR&D is a really steep venue to get to talk about organic chemistry in. And so recall that machine learning is just data plus descriptors plus algorithms. So I just wanted to talk very briefly in this context about ChatGPT. I think I already said what I wanted to say here is that ChatGPT has the data of the internet, and it uses billions of descriptors if we're talking about ChatGPT 3, which is what we used for our experiment, trillions of descriptors if we're talking about ChatGPT 4. And the algorithm is called a large language model, or LLM, but there's obviously a lot of nuance to that OpenAI would have batched up as proprietary. But anyways, we went to ChatGPT and we said, dear ChatGPT, generate a 3-ligand by 4 catalyst by 2 base reaction array to optimize Buchwald-Hartwig coupling, and give us the data as rows and columns, include specific recommendations for other reaction parameters, respond succinctly, finally cite some literature, now, this is the answer that it gave. This is a fine answer. And so I imagine that a lot of people on the call are not used to thinking about matching catalyst to Buchwald-Hartwig coupling conditions. Historically, I have been handsomely paid to go to pharmaceutical companies and give them recipes like this. But now with the ChatGPT prompt, it's quite straightforward to get the recipe yourself. And so XPhos, SPhos, and RuPhos are ligands. They are phosphine ligands that are very reasonable proposals to run a Buchwald-Hartwig coupling. And these bases are reasonable. These catalysts are reasonable. The temperature, the solvent it proposes, the reaction time, everything is totally reasonable for ChatGPT to propose. The only thing we gave it was this block of text, right? And it went and understood to a certain degree the chemical literature. It doesn't understand the mechanism. It doesn't understand where atoms are. It doesn't understand the physics of chemistry. It just understands that lots of people have published papers on the Buchwald-Hartwig coupling with these types of ligands. And the beauty is that we plugged it into our software Factor, which builds combinatorial arrays. And so we are not telling, hey, ChatGPT, give me the exact answer. I think that ChatGPT would probably not do so well there. We said, give us a ballpark answer, and we'll build an array of 24 possibilities around that. And so our Factor software gives us this array. The pink and yellow lines show the recipe of different reagents that ChatGPT recommended we put in, and then the output is shown as this blue and yellow heat map. With a yellow dot here. We actually scaled up this hit with palladium DPPF and RuPhos and K3PO4, and the crude reaction trace is shown here. So there's an internal standard which we added. There's product, and this little other blip is the ligand itself. And so this is an incredibly pure chemical reaction. It is we isolated, I think, an 81% yield. And so we were very pleased with the first chemical reaction that we're aware of that was run on with ChatGPT's help. Now, I talked a lot about amine acid coupling reactions and about clicking together virtual chemical spaces, and everything there that we talked about was single step reactions. So can I take an amine and acid and make an amide bond? Can I take an amine and acid and make a carbon-carbon bond? It's all a single step, and that is the bread and butter of much of the pharmaceutical industry. But to get to molecules like this, we're going to need to move into multi-step synthesis. And I just want to quickly close with our recent entry in multi-step synthesis, where we were interested in this particular natural product that comes out of the stomona plant, which has been used for lung health in Chinese traditional medicine for about 300 years or more. And the interesting thing is that this molecule had been synthesized 32 times before. And so there was a large history to how this molecule had been made. We plugged it into a different software and achieved the following synthesis, where we buy this lactol, we do what's called a brown alylation, we install the cyclobutanone, and then add a hydroazidation reagent and work up with TICL4. The synthesis is done in 3 steps, and this is just a really short synthesis of this particular molecule, and so we're very excited by this work. And so here's what I hoped to cover today. really I think the thing that I hope people will take away from the talk is that machine learning is just a set of data mapped to a bunch of descriptors and then some algorithm that navigates through it. And I'd like to leave you with this question: does synthesis really matter? I think, I hope that I've convinced you that it is an important part of our endeavor. It's my great honor to work with such a wonderful, incredible team. You guys make it fun, and we laugh a lot. And we've been able to grow the lab very well under your guidance. So I thank you so much for this opportunity.
Online. Thank you very much. Now I understand what you're doing. I just heard of the machine learning, the chemical things. I don't know exactly what you're doing. Now I understand some. So while any online question or audience, while they are preparing, I ask a question. In old days, one professor can invent one reaction for the whole life, then they name that reaction. What are the reaction you name? Did you name the reaction?
Oh, yeah, the Cernak-Shen reaction is the esterification of amines and acids.
Yeah. So any question? Yes, Dan.
You came from pharmaceutical industry, and it seems like they would have a lot of financial incentive to create these reactions, so they have much better ways to build new molecules. Why is there a lot of this kind of work going on in the pharmaceutical industry? And if not, why not? Or is it possible that they have other reactions that they don't tell the world about because they want to keep them private and confidential?
Yeah, I love that question, Dan. Thank you. The question is, to what degree do they invent new reactions in the pharmaceutical industry? I think that today there's maybe a little bit of that happening, but I think realistically there's very little. It's really the it's our marching orders in academia to invent these new chemical reactions. There has been a little bit of work in that space, but I tried to show some like modern drugs, this PCSK9 inhibitor and the sotorasib one. I mean, all of these would be on the basis of academic publications where there's a new chemical reaction that made that bond arrangement possible today. And it's through partnership between academia and the pharmaceutical industry. I think that there is a interest in advancing known chemical reactions in the pharmaceutical industry. We tend to publish on simple molecules, and I showed how we ground up pills to kind of like work with more complicated molecules. A lot of the reactions that we develop on simple molecules, you bring them to the pharmaceutical industry where they're necessarily working with really complex molecules. And we did it on something that had like 8 atoms over here and 10 atoms over here. Well, they have like 50 atoms, and those atoms like poison the catalyst that we put in. So a lot of the reactions we develop in our labs require advancement to be used in the real life setting. And so they might do some of that work, but realistically, they just read the modern literature and say, all right, here's a new reaction, I'll try it. They're deeply invested in making new chemicals, just kind of piggybacking off the reactions that are there.
Don't like question.
Oh, great.
Put down. Put down. There.
Right. So are you familiar with the pharma AI platform in silico? What are your thoughts about? Yeah. So, okay. So yeah, so in silico is a company. I guess I realized I didn't really talk too much about where the pharmaceutical industry is right now. So in silico is definitely one of the leading companies in terms of applying AI for chemistry. I would point to a lot of other leaders, Octant and Relay and Entos and Exscientia are probably other big players in this space. I mean, all right. So in silico made a big splash with a inhibitor that was then criticized as being, yes, generated by AI, but only having an extra single carbon atom added to it. It took an amide bond. There was an amide in the molecule that they took as the and they closed it into a ring by adding 1 carbon atom. And so yes, it was invented by AI, but if you held it up to a human medicinal chemist, they would say, yeah, of course, right? Like, of course that that's not surprising. I do think that in silico has advanced quite significantly. And one thing I think that they're doing that's really clever is they have, you know, I showed this notion of design, make, test, design, make, test. They are doing a lot of that with robots, as are many companies. We're, you know, and as is our lab. I do think that In Silico is doing some really great things. I do think that In Silico and other companies are about to enter a step change. I think that in the next 10 years, it's going to be really easy to invent a new small molecule inhibitor if you know the protein target that you want to go after. You don't need to know the protein structure, right? Just its name or some association with disease. We'll be able to generate the protein structure. We'll be able to generate small molecules in the computer and then quickly on robotic platforms later. I think that, you know, we will soon be entering a phase where precision medicine is a lot more in reach than it is today. Because it'll just be cheaper and easier to invent new medicines. Because the synthesis I've been talking about is like 200 years old, or so there's such a long history that there's because it's based on graphs. You can plug in 200 years of mathematics behind this. It's just there's been some experimental challenges we've been overcoming, but I think over the next 10 years we're going to see a big step change.
To follow up with that question, the Silico is that they published the Nature Biotechnology for the DDK1 paper.
Right.
So they argue AI just generated 6 compounds, they synthesize 50% of their works, right?
Right.
I see the value. Based on your information, how many big pharma already doing similar things like they do today?
Yeah. So the question is, how many big pharma companies are using predictive? Like 100% of pharmaceutical companies are using predictive modeling in the discovery of their drugs. Because, you know, I mean, I mentioned a couple times that it's a $1 trillion industry. We fail all the time, right? I mean, like you just said, like if we could predict whatever the number was, 600 compounds, 50% of them are active. That's a huge number, right? I mean, I would say, you know, probably a lot of people would be happy with a 5% success rate. The success rates are really, really low for inventing bioactive inhibitors, right? Just does it bind to the protein? That's still quite difficult. And our point is that it's actually very difficult to run chemical reactions to make the thing, but you don't get any of that information in the literature, right? So if you read the literature, you have the impression every chemical reaction works. We can show you, I don't know what, 50,000, 100,000 reactions that don't work. And so, you know, so the pharmaceutical industry fails a lot. They know this is not published. It's a $1 trillion industry. So they are eager to find a way to fix that using data predictive algorithms.
Any other questions from the audience? I have one more. Well, the data you showed, the AI data, or they showed, what is the minimum number of compound? You say, hey, AI I believe their prediction is okay. There's a cutoff, sort of.
Oh,
to train the AI say, oh yeah, I can make a reasonable prediction.
Yeah. So typically what we would use is what's called a receiver operating curve, and it's just a straight line. It's like an X by Y plot with a straight line, and that straight line is random guessing. And your goal is to have a curve. Your data falls on a curve that goes just a little bit above that line. It means that your algorithm is better than a random guess. Is the typical way that we would test. And so, and that's the marker, is like, you know, that your algorithm is working if it's better than random. And you know, many times it's worse than random, right? And so you have to be careful about that.
Any other question online? No? Thank you very much. Thank you, people logging online and in person. Thank you. Thank you, Tim.
Thank you. Yeah, I mean, I'm.
Yeah.
Yeah. If, how,